Okay, hello everyone, and welcome to another session of the Anatis Knowledge Academy webinar series. Today we are pleased to have Morgan with us sharing her knowledge and expertise on artificial lighting in greenhouses. Morgan is a PhD candidate in plant biology at the University of Laval. She applies her entomology background to improve pest control in greenhouse crops. With almost 10 years of experience in biocontrol, she studies insect behavior and plant protection. Morgan actively collaborates with biocontrol companies and growers across Quebec to develop practical solutions. So thank you so much, Morgan, for accepting our invitation, for sharing your knowledge with our attendees today.
Thank you very much. Hello, everyone, and thank you for having me in Anatis Knowledge Academy. I'm quite excited to dive into this topic today because while we often talk about lighting in terms of plant growth, there is a whole side of the story that's still in the shadows. And today we will bring the bugs into the spotlight. You may be familiar with the benefits of LEDs for plant growth, especially in controlled environments like greenhouses, where light spectrum, intensity, and photoperiod modulation are now routinely used, whether for seedlings, ornamental crops, or to boost yields. But lighting doesn't just affect plants. It also shapes the tiny world of insects around them. Thrips, aphids, whiteflies, and even their beneficials all react to the lighting conditions. Their behavior can shift according to lighting, and this can make or break your biocontrol success. Today, we will explore how to turn light into an ally, not just for your plants, but for your pest control too, with a special focus on beneficial. I will start with a quick overview of how insects perceive light, just enough to understand the basics. Then I will share a few selected examples of how lighting can promote biocontrol. Finally, we will take a closer look at new findings regarding the predatory bug 𝗵𝗿𝗄𝑆 insidiosus. My goal today is to share scientific insights that bring insect ecology into focus and show how lighting can be used more intentionally to support biocontrol. To begin with, insects aren't just passive under greenhouse lights. Their vision actively shapes how they behave. So, how does it work? Here is the basic structure of a compound eye, which is very different from ours. Instead of having a single lens like a human eye, insect eyes are made up of many tiny repeating units called ommatidia. You can think of them as tiny light sensors arranged in a mosaic, giving insects a wide angle and pixelated view of their environment. Each ommatidium has its own cornea and crystalline cone that focus light onto a set of photoreceptors. And the central light-sensitive part, here in blue, is called the rhabdom, where light colors and brightness are detected by visual pigments. These signals are then sent to the insect's brain through the axon. The number and arrangement of omatidea vary by species, depending on their lifestyle. For instance, ladybugs and lacewings are predators, so they rely on good motion detection and contrast to spot their prey. In addition to their large compound eyes, many insects, like wasps and hornets here, also have simple eyes, called ocelli. These don't form detailed images, but instead detect changes in light intensity. Each ocellus has a single lens and a layer of light-sensitive cells beneath. So it's much more basic than a compound eye. As for mites, whether pest or beneficial mite species, their visual systems are less sophisticated than those of insects. Some mite species might have ocelli or extra retinal photoreceptors, so they may be still sensitive to light intensity and photoperiod, but they rely much more on chemical cues for navigation. That's why their responses to artificial lighting are less studied. Altogether, compound and simple eyes allow insects to detect incredibly subtle variations in light, a skill so razor sharp it helps mosquitoes dodge your hand in a split second. So insect vision is remarkably efficient. More specifically, when looking at the light spectrum here, most insects have what is called a bichromatic vision, with sensitivity peaks in the UV blue and green ranges. In contrast, plants primarily absorb blue and red wavelength for photosynthesis, which is why blue to red ratios are a key component in horticultural lighting. But insects don't just detect light, they decode it. Light guides their movement, feeding, mating, and internal rhythms. And in greenhouses, LEDs can shift all those cues. My key message here: insect vision is real and seriously sophisticated. Early in my PhD, I stumbled on this study, where researchers built a full 3D cinema portraying mantises, even with tiny little glasses, to test the predation behavior of praying mantises. And it turns out that mantises use stereoscopic vision, just like us, to judge depth. So it's a funny but quite striking example of how finely tuned insect vision can be. So yes, lighting shapes insect behavior. But the real question is, how can we turn that to our advantage? So far, most optical studies have focused on pests, aiming to disrupt behaviors like orientation, feeding, or reproduction. However, responses can vary wildly, and I mean wildly, for both pests or beneficials, depending on the species studied and the lighting conditions. So any recommendations should be applied with caution. The next example highlights just how much lighting conditions can influence the outcome of biocontrol for a same species. The green peach aphid is a widespread generalist pest in greenhouse crop. And its control strategies have already been addressed in previous sessions of the Anatis Knowledge Academy, if you are interested. So in one study, researchers tested whether UV-enriched artificial light could help trigger plant defense pathways in order to reduce aphid populations. And in another study, different horticultural lighting conditions were used to extend the photoperiod with LEDs, simulating typical winter greenhouse settings. But could this longer day length also unintentionally boost aphid activity? So these are two distinct trials using different light treatments, one aiming to reduce aphids indirectly through plant defenses, and the other raising concerns about potentially favoring aphids under extended light exposure. In the first example, plants and aphids were exposed either to fluorescent light alone, the control treatment, or to a mix of fluorescent and UV-enriched lights. Surprisingly, aphids reared under UVA and UVB conditions had significantly higher body weight and fecundity compared to the control, even though their development time didn't change. So despite shifts in plant chemistry in response to UV light, these UV conditions turns out to favor aphid outbreaks rather than suppress them. In the second study, things look quite different. This time, in growth chambers again, researchers tested whether light spectrum or photoperiod duration could influence aphid reproduction. And they found no effect at all, whether plants were under 12 or up to 20 hours of light, aphid reproduction stayed the same. And this held true whether the light was full spectrum white, red-blue, or a narrowband blue-green-red mix. So in this case, lighting could be tailored to plant needs without the risk of boosting green peach aphid populations. As we've just seen, even for a single pest species, responses can vary depending on the exact lighting conditions. And with LEDs allowing us to tweak spectrum intensity and photoperiod, the number of possible combinations is nearly endless. But despite that complexity, some patterns are emerging, especially when it comes to disrupting the ecology of major greenhouse pests using targeted light spectra. For example, the whitefly's proliferation under artificial lighting was a major concern when LEDs were first introduced in horticultural settings. And nowadays, evidence strongly suggests that the use of blue light in combination with the use of new LED, the development of new LED trapping techniques can successfully control them. While we must avoid excessive green and yellow component in ambient greenhouse lighting. for a deeper dive on specific responses of pest or beneficial species and recommendations for lighting applications regarding biological control, I recommend these excellent references. So these aren't the only studies out there, but in my opinion, they give a clear and useful summary of the main takeaways. And before we go on with our second example, I would like to take a moment to mention two optical manipulation tools for pest management that are really gaining traction for greenhouse use, starting with new LED trapping techniques. On the research side, we now have species-specific traps, combining sticky cards with blue or green LEDs. These traps are fine-tuned to match a specific pest visual sensitivity and have shown promising results, as with whiteflies. That said, your good old yellow sticky traps still do a great job because they target the green light sensitivity peak, which works well for most pest species. On the commercial side, generalist LED traps are also available, often using broad spectrum or black light LEDs with an effective range of up to 40 to 50 meters. But here is a catch. They are so attractive to insects that they should only be used at night to avoid catching bumblebees. They also work best inside the greenhouse during winter only and outside during summer, when pests are more active and reactive to light cues. On the other hand, UV blocking materials have already received a lot of attention in recent decades because UV light is a key cue that insects use to orient themselves during flight and choose where to land. So, by filtering out UV radiation, these covers can disrupt that orientation process and reduce insect landings. Greenhouse studies show a significant decrease in aphids, thrips, and whiteflies, and even reduced virus transmission by these pests. But not all covers are created equal, and recent work by my colleague Andréane Couture, right here at Université Laval, tested UV blocking films currently available in Quebec. And surprisingly, some covers led to more aphids and leafhoppers in buried tunnels. So once again, it's a great tool, but it needs to be carefully chosen and adapted to both your pest pressure and crop type. So far, we've seen how lighting strategies, and especially spectrum, can help reduce pest pressure, either by disrupting their behavior or by enhancing traps and covers. These are powerful tools, but as Johnson and colleagues pointed out, we still know far less about how light affects beneficial insects, especially those we release in greenhouses. Until recently, most efforts focused on using visual cues to attract wild beneficials from surrounding habitats into crops. But what if we could go further and use light to actually boost the performance of beneficials already at work in our systems? Well, that's exactly the matter in this next example. Let's take a look at two closely related parasitoid species, Aphidius matricariae and 𝗵𝗿𝗄𝗃, both widely used in commercial aphid biocontrol programs. These tiny wasps lay their eggs inside aphids, eventually killing them and producing a new generation of wasps. So, can we use longer days and tailored LEDs to give these wasps a real performance boost? And just as importantly, what spectrum works best? As you will see, each species may have its own lighting preferences, highlighting the importance of evaluating responses on a species-by-species basis. Let's start with 𝗵𝗿𝗄𝗃 matricariae. In greenhouse tests, plants, aphids, and parasitoids were exposed to 12 hours of natural sunlight, extended with either blue-red or blue-green-red artificial light, no extension for the control. Parasitism remained consistent across all conditions with no differences due to photoperiod or spectrum. The presence of wasps significantly reduced aphid numbers, shown in blue, compared to cages with plants and aphids only in yellow. So, this parasitoid's control capacity was unaffected by lighting conditions. 𝗵𝗿𝗄𝗃 matricariae did a great job no matter the lighting. Now, let's turn to 𝗵𝗿𝗄𝗃, a species with a different story. In growth chambers, plants, aphids, and parasitoids again were exposed for 8 hours to what the authors considered as the solar spectrum, with or without extending the photoperiod with different blue-red LEDs ratio. The probability of parasitism increased by 20% under LED lighting, and significantly more eggs were found at the end of the day exposure, I will go again. So more eggs were found after extending the photoperiod compared to the end of the day exposure. Okay? So this indicates here a real interest in extending the photoperiod to support the parasitoid's control activity. Considering now the different spectra used for extension, we note no difference except in the case of exposure to 100% red light, where parasitism is significantly reduced. Overall, this finding regarding 𝗵𝗿𝗄𝗃 under artificial lighting are encouraging, but an additional aspect remains to be considered. The spectrum also affects the sex ratio of emerging wasps. Under light, 80% of the second-generation parasitoids are males, but only females control aphids by laying eggs. So the practical takeaway here is that too much red light can cause a sex ratio imbalance for 𝗵𝗿𝗄𝗃, potentially reducing pest control, but also reducing parasitoid reproduction over time. By the way, this strategy of extending the photoperiod by adding specific light spectra has shown positive effect on several other beneficial species, mainly boosting egg laying and hatch rates, speeding up their development, and ultimately improving parasitism or prey consumption. Historically, Parasitoid wasps have been more extensively studied than predators for their responses to lighting, as they are known for their particularly sensitive visual system. On the other hand, recent evidence shows that predators are also sensitive to artificial lighting. And I can't resist sharing our current work on the predatory bug 𝗵𝗿𝗲𝗄𝗃 insidiosus, the minute pirate bug. My mission was to discover the ideal lighting ambience for them, ensuring that they would have kind of a perfect first date, enjoy a nice meal, feel in the mood for reproduction, and ultimately raise a thriving predator family to ensure an effective biocontrol. 𝗵𝗿𝗲𝗄𝗃 has been a key beneficial in North America for over half a century. It's particularly effective against western Florida thrips from 𝗘𝗃𝗲𝗏 occidentalis, a pet peeve of growers in both vegetable and ornamental crops, as highlighted in previous sessions of Anatis Academy. We faced the research imbalance I mentioned earlier. While there are at least 41 studies on thrips visual ecology, only 7 focus on 𝗵𝗿𝗲𝗄𝗃 insidiosus under artificial lighting, and these are limited to manipulating photoperiod to address winter diapods. Expanding to other 𝗵𝗿𝗲𝗄𝗃 species, it's clear that their orientation, dispersal, locomotion, and reproduction are also influenced by light spectrum and light intensity. However, we had no information regarding 𝗵𝗿𝗲𝗄𝗃 insidiosus, despite this species being widely used under artificial lighting, and the potential of horticultural lighting to enhance its predation was unexplored. So our research team has focused on this gap, proposing an initial experiment to identify the optimal lighting conditions for 𝗵𝗿𝗲𝗄𝗃 predation. Our goal was to test a range of spectra combined with varying light intensities. We used monochromatic blue-green red, as well as some blue-red ratios, and a blue-green red mix based on Garcia and Lopez recommendations for optimizing seedlings growth of greenhouse crops. Predators were placed in arenas with 5 thrips, and their behavior was recorded for 5 minutes. What did this look like in practice? We focused on 3 key predator behaviors: walking, attacks, and captures. Here, the predator at the top of the petri dish quickly attacks the nearby thrips on the left. The prey escapes, but the predator moves on and soon captures the thrips at the bottom right. Prey is immobilized, now dead, and the predator begins to feed. This is a successful capture. We were interested in different variables regarding predator-prey interactions, as does the predator attack quickly, how much time was spent walking, etc. And we found that the light spectrum significantly influenced all recorded behaviors, while light intensity had negligible effect. All the details can be found in our article here. For now, I will just mention our main finding, which was the strong effect of spectrum on successful captures. While predators could capture under all tested conditions, in the 10% blue, 90% red treatment, the addition of a small amount of blue doubled the success rate compared to the red light alone. So based on these results, we selected 3 light treatments for further experiments to test these trends with performance indicators now related to mating and development of organs. While this initial experiment used a custom optical device for high-quality imaging, the next steps were conducted in growth temps. In the upcoming experiments, insects will be exposed to 12 hours of an artificially recreated solar spectrum with sunrise and sunset phases mimicking a cloudy March day in Quebec, followed by 8 hours of one of the 3 previously identified spectra, except in the control treatment without light extension. First, we compared mating behaviors across the 4 tested spectra: sunlight, blue and red, and blue. After being acclimated to the tested light sequences, Orioles couples were given 15 minutes of relative privacy. Here you can see the female at the bottom and the male at the top of the screen. The male makes a timid approach, but then contacts the female. Marking a mating event. So moving on with mating probability. Mating probability showed no significant differences between treatments, though there was a trend of fewer matings under blue light, while the other treatments hovered around 60%, with blue-red light having the highest probability. This trend with blue light is also observed in the number of mating events and their total duration. These two parameters, mating frequency and mating duration, are important indicators as they directly influence the female's fertility. More frequent and longer mating typically leads to a larger offspring. This brings us to our next experiment, focusing on the development of organs. The couples were exposed to the light sequences for 48 hours, and egg laying was monitored in females for 6 days. The eggs were then incubated under the same light treatment to track development until the emergence of the new generation. We observed a significant effect of the extension spectrum on fertility. Surprisingly, the blue light treatment resulted in the highest number of eggs, nearly double that of the control. As for the proportion of eggs that hatched and produced juvenile predators, the control showed a lower development compared to the 3 treatments with photoperiod extension, especially blue and blue-green-red spectrum. Thus, the number of juveniles after 2 weeks followed a similar trend. Blue light led to a higher number of predators, while the control treatment resulted in lower development. when looking at the proportion of offspring completing their metamorphosis into adults, this time the highest value was observed in the control treatment. Although, by the end of the development cycle, we don't ultimately see a significant differences in the number of adults produced between treatments. However, the blue light treatment produced significantly more adults than the control. Finally, the proportion of females in the offspring, a crucial factor for population viability, remained above the critical 45% threshold set by Anatis Bioprotection. This ensures there would be more than enough females to support a third generation of predators, potentially sustaining ongoing pest control. So to recap, we found that blue light is associated with fewer and shorter mating events. This aligns with previous studies on 𝗵𝗿𝗄𝗏, where both the likelihood and duration of mating were reduced under blue light. When we extended the photoperiod with blue light, it resulted in the highest number of offspring, supporting findings on the fecundity of 𝗵𝗿𝗄𝗏 insidiosus under low-intensity blue light. We also observed that blue light favors egg laying and hatching, but not metamorphosis, confirming that the effect of blue light on insect development are highly dependent on both species and life stage. Curiously, despite reduced mating under blue light, we still observe higher fecundity, fertility, and more adults in the second generation. We suspect that mating stress plays a role here, making the less mating promoting spectrum more beneficial for population growth. Traumatic insemination, where the male pierces the female's body to deposit sperm, has been observed in the 𝗿𝗂𝗏𝗏 family and likely occurs in 𝗵𝗿𝗏𝗏 insidiosus. While necessary for reproduction, it can reduce female longevity and fertility due to energy loss, injury, or infections. So after considering independently predation, mating, and now development, we wanted to see how our light sequences impact the whole plant pest predator system over time. So the next part of our experiments explores how light exposure shapes biological control over several weeks. Plants, pests, and predators were exposed to tested light sequences for 2 weeks. On the left graph, predator densities remain consistent across treatment with just a slight dip in 𝗵𝗿𝗄𝗏 number under blue, green, red compared to the control. But what's striking is a significant drop of trips numbers when predators were present in blue versus in trips only condition in orange, no matter the lighting condition. So overall, 𝗵𝗿𝗄𝗏 delivered strong control over every lighting sequence. And on another positive note, we also saw a clear reduction in trips damage, again regardless of lighting, the presence of 𝗵𝗿𝗄𝗏 cut leaf damage by 46%, which really confirms their effectiveness as biocontrol agents under all tested conditions. Interestingly, plant fresh biomass was higher when predators were present, somewhere between the levels seen in trips free plants or those with trips. And photosynthetic performance was modulated by light sequence with the best results under blue and blue-red lighting as expected. We are currently digging into how light and insects together shape plant hormones with the help from my accomplice, Dr. Adam Barada, since lighting can also affect insects indirectly through the plant. To sum it up, the blue-red spectrum gave us the best predation results, while pure blue led to fewer captures. Interestingly, blue light reduced mating rates but boosted offspring development, probably tied to mating stress and traumatic insemination, as we mentioned earlier. When we looked at the full plant pest predator system, those treatment differences disappeared. Aureus maintained solid performance under all lighting conditions during extended plant exposure. That's why it's so valuable to start with lab trials before scaling up in real settings. And we are pleased to report a successful Aureus establishment and control against thrips last winter in a commercial strawberry greenhouse under artificial lighting. Our partner, 𝗵𝗿𝗶𝗆𝗿, even resumed winter releases thanks to these results. And we will be publishing the full story soon in 𝗿𝗏𝗏 Daily, so keep an eye out. Based on these results, here is what I would currently recommend. For mass rearing, the photoperiod extension with blue light would be good during early development to reduce mating stress and enhance egg laying and hatching. This should be followed by a transition to low spectrum light resembling sunlight to promote adult metamorphosis. In greenhouse settings now, a 10% blue, 90% red light extension at the end of the day could enhance predation, aiding in pest outbreak prevention during at-risk periods, for example. Additionally, photoperiod extension with blue light combined with natural solar radiation is likely to promote Aureus symbiosis growth and establishment after predator introduction in greenhouses. To wrap up this Aureus example, I wanted to ask one last question. Could their response to lighting vary depending on their biological status? After all, when we order from Anatis, we receive a mix of Aureus females, males, and juveniles. Differences have already been reported in other beneficial species, but for Aureus, only females are typically considered in trials. It's often assumed they spend more time hunting and contribute more to pest control, while males who have shorter lifespans focus more on courting females than catching thrips. So I used my growth tents and a camera setup to directly compare the predation abilities of females, males, and juveniles under our different lighting treatments of interest. Indeed, as expected, females captured the most thrips overall. But interestingly, males and juveniles still contributed with up to 38 and 33% capture proportion, respectively. Statistical tests didn't reveal any significant differences between the light spectra, so lighting didn't seem to impact predation performance across groups. What stood out, though, was the behavior. Females tended to be more stationary. Once they cooked a tray, they would stop to enjoy their meal. In contrast, males were by far most active. We often observe them dashing around the arena almost frantically. This hyperactivity probably increased their chances of bumping into a prey, which may explain why they consistently showed a high number of attacks here in blue. Males were maybe searching for females, but that behavior might actually work in our favor. Because while they are zipping around, aureus can trigger some sort of psychological warfare with threats. This has already been described in other studies. Even without direct attacks, the simple presence and movements of aureus on a plant can stress thrips enough to reduce their fitness. And here is what I find exciting. Our results suggest that every aureus you receive from your supplier is doing something for biocontrol under artificial lighting. Not just the females, but also the males and even the juveniles. Before concluding, just a quick glance at other predator species. Efforts have been made to explore their responses towards artificial lighting, but further research is still needed for most of them. Notably, the American 𝗷𝗶𝗴𝗵𝗁 stands out as a promising candidate for Quebec and Ontario, combining fault tolerance with efficient reproduction under LED lighting in greenhouses. Finally, here are the key takeaways. Light clearly influences insect behavior and rhythm. While pests have been studied more than beneficials, we can already use lighting to disrupt their activity. But responses are species-specific, so it's important to test carefully before applying in real settings. There is no such thing as a one-size-fits-all magic lighting recipe. The results show that beneficials like 𝗵𝗵𝗄𝗏 and aureus can maintain strong performance under LEDs, and in some cases, specific spectra may even enhance their impact. But more research is needed to tailor light strategies for key species in commercial greenhouses. Before we move to the questions, I want to take a moment to sincerely thank my fantastic partners. This work would not have been possible without the technical support from Anatis Bioprotection and Solum Technologies, as well as the collaboration with 𝗵𝗿𝗏𝗄𝘆𝗀 horticulture, who grounded our work in the reality of commercial production. And thank you all for your attention today. I'm now happy to take any questions in English or French.
Okay, thank you so much, Morgan, for such a great and informative presentation. So if anybody has a question, just let me know, raise your hand. I will give you access to mic and you can just directly ask your question. So I couldn't see any question, but I would like to ask you about maybe can you let me know or for the so I can so first I will give the access to Samantha, then I will ask you my question, my own question. So Samantha, go ahead. Hello, Samantha. Can you hear me?
I can't hear anything. I can't hear anything for now, Samantha.
I also couldn't hear, but I don't know. I already gave her both camera and mic.
Oh,
Can you hear me now?
yes.
Yeah, it's okay now.
Oh,
I'm Hello,
so
Samantha. Yeah.
sorry. Hi, I was
No
having a mic
worries.
issue and okay. I had a question about the study that you referenced that found that aphids benefited from plants exposed to ultraviolet radiation. I think there might be differences between UVA and UVB, I assume. On plant chemistry, one's a lot more intense than the others. Can you resolve that for me? Do you know what type of UV those plants were exposed to in that study?
Yeah, in fact, there were two different. There was one kind of UVB and two kinds of UVA, and I guess one treatment that the authors referred to as violet light is at the beginning of the visible spectrum. So I think they choose these specific wavelengths because of the plants they studied, not because of the insect, but because of the plants, because these teams, they are plant chemists. So they were working on broccoli for the study I mentioned today. And I think they were really interested in the intricate chemistry things between UV light and broccoli plant reactions, and they only have a few portions of their study on aphids. Does it respond to your question, or?
Yeah, it does. It gets me started. I will definitely take a look at that paper more closely. Thank you very much.
Thank you,
For your
Samantha.
question.
Yeah. Okay. So, any question? So you know that Morgan, one of the most important thing I think for growers and for farmers about the establishment of Oreos.
Yeah.
And so what do you think about how many hours of light is necessary at least for establishment? You know, for example, okay, we gonna to establish or start releasing the Oreos in our farm. What do you think? For example, 12 hours at least we have, or 10 hours at least we have. How many hours at least is necessary?
It's a very well documented question, but I know Oreos is tricky under winter conditions in greenhouses. At least 10 to 12 hours of light exposure, whether the spectra, it will help for avoiding diapause. Okay, so this is the first thing. But if you want to not just avoid the diapause, but also promote its establishment and its reproduction, we've shown that maybe using a specific blue light to do at least a part of this 12 hours lighting exposure could be a good idea.
Oh, perfect. So thank you. So Ben has a question. I will give access to mic and camera. Hello, Ben. Go ahead.
Hello. Thank you to the both of you for helping to organize this event. My question is, has there been any research regarding the difference between rearing Aureus under LEDs versus high pressure sodium lights or metal halide lights?
Yeah, I think in fact in previous studies, in previous time, where HPS were the most used lighting sources in greenhouses, there have been some studies on mainly photoperiod views to alleviate diapause in Oreos. But I don't remember of any direct comparisons between HPS and LEDs, actually. And I would be really interested in doing that because I think lots of growers still use HPS. So yeah, I will definitely think about that.
Oh, great, thank you very much.
Thanks, Ben. Oui, Nicolas?
Okay, so we have any other question? Okay, so I can see here Nicholas. I will give him access.
Okay,
Hello,
can you
Nick.
hear me?
Yeah, hello, Nick. So go ahead.
All right. Hello, very great study, by the way. I have a little question here about the studies you've been reading on the Aphidius.
Yeah.
So I have a grower that I know that I'm working with who has a problem with aphids at his greenhouse, and he's trying to breed Aphidius 𝗂𝘃𝗏 or 𝗴𝗿𝗏𝗏. I can't remember which one of them it is, but do you have, in regards of what you've been reading, do you have any cues that could help him maybe in his breeding about the light spectra that could be used to promote the reproduction of Aphidius, or have you been reading anything about that? Because he can't actually, he's working with HPS in his greenhouse, and he can't change his light spectrum. So maybe if you know anything that could help that you've been reading.
This could be due to other aspect, but what I can think about really quickly, I don't know the greenhouse, I don't know the setup, and I don't know the exact conditions, but sometimes HPS, we have too much red light component in it, and some species of Aphidius really doesn't like that. Like Aphidius 𝗧𝗏𝗏, as I've shown, the sex ratios can be biased, okay, with too much red light. So maybe this is a part of the response here. I will try, maybe Omid correct me if needed, but maybe just having a more regular Aphidius releases, because if the problem is that they couldn't reproduce or that the sex ratio of the second generation is biased, and you tell me that he's working with only HPS lamp, that will be the only solution I can quickly think about for now.
Okay, because his predators are working well in the greenhouse, but it's mostly about the breeding. If I wanted to know if you had any cues, because maybe the changing his light spectra in his other room in which he's trying to do his breeding right now could have helped, but it's fine. I'll.
Okay, so if I understand correctly, he has two separate lighting setup. He has the greenhouse with HPS lamps, and he had like LED settings in.
Yeah, he has another room in which he can plays with the LEDs and the spectrum. So maybe, yeah. So I was thinking maybe you would know something about the light spectra that could promote the reproduction of or help in his breeding of the Aphidius species he's using. But yeah, I think it's 𝗂𝘃𝗏𝗏 that he's using right now.
Okay. So as we've seen today, it's really important to know exactly the species of Aphidius he is using, okay? Because we need to adapt accordingly the light spectrum. But I would definitely recommend to him to read the two papers of 𝗵𝗫 𝗂𝗏 that I mentioned today, and that were done at Laval University also, by the way, a little promotion for our university. But I think the responses are there, and Priscilla have shown very clearly that Aphidius 𝗧𝗏𝗏, for example, could be used under a wide variety of LEDs lighting environment, but it just don't like too much red light. So maybe this is the first steps to tailor the perfect breeding environment for your producer. But I will definitely be curious about talking a bit more about this question. So if you want to let me know what happened or what he tested, I would be curious to know.
Yeah, sure. Thanks for your answer, and I'll keep looking, and I'll write back to you probably on LinkedIn. Thanks for everything.
Thank you, Nick. Thank you. So another question for Carling. So Hello.
Yep, hello, can you hear me?
Yeah, I can. Go ahead.
Hi there. It was a great presentation. I just had a question about the, you had mentioned that this winter you had successfully established and had thrips control in a strawberry greenhouse. I was just wondering about the details of the lighting that you used. Was it the blue-red with the extended photo period?
As I said, we are currently preparing a whole article that we will put online on the magazine 𝗫𝗏𝗏 Daily, the online magazine 𝗫𝗏𝗏 Daily, with the whole details of the lighting setup and stuff like that. But it was the lighting setup was LEDs, and it was very close to one of the treatments we used for Aureus in the lab. So stay tuned, and you will have all the details really quickly.
Okay, awesome. Thank you so much.
Thank you so much. So, okay, so is there any other question here? I couldn't see. So, if there is no question, thank you all for attending today's webinar. We appreciate your time and good questions and good conversation. We hope you found it useful and look forward to connecting again soon. Thank you so much and have a good one. Thank you, Morgan.